Hey Lisa, do you know how you can't tell if a pterodactyl is going to the bathroom?
Right.
Because the P is silent. Ah.
Get
Okay. Hey everybody, and welcome back to week 5 lectures for Geology 309, Bringing Fossils to Life. We're going to be wrapping up our first unit of lectures and notes in preparation for our upcoming exam. And the last set of lectures today are going to be over biostratigraphy and biogeography. So we're going to start out talking about biostratigraphy, which is essentially how we use fossils to help us determine relative age. And if you've had a couple of basic geology courses yet, which hopefully all of you have, you'll be familiar with this guy, Nicholas Steno, father of modern stratigraphy. So we're going to review some stratigraphic concepts and then get into the details of biostratigraphy. So let's jump right in. First thing first, let's get some terminology down. Stratum, a stratum, or the plural strata, is a distinct, relatively continuous, widely deposited rock layer defined by common lithology or rock type. So any layering that we're seeing predominantly with sedimentary rock, but with any kind of rock, would be stratum. If there's numerous layers, then it's strata. This may or may not be an isochronous unit. Now, if you're not familiar with that term isochronous, breaking it down, iso meaning one, chronos referring to time. So what this means is you can follow a stratum over some distance, and it may not all represent the same equivalent time. Okay, especially if we're just looking at rock layers. Thicknesses may vary spatially. You can have a stratum that starts out 10 centimeters thick, and a few meters later it can be a meter, meter and a half thick. That doesn't mean that the uppermost and lowermost boundary of that stratum are all the same age. So just something to keep in mind. The study of stratum or strata, the rock layers of the Earth, is stratigraphy. So there's an entire course of stratigraphy and basin analysis taught by yours truly that I encourage you to sign up for in the upcoming spring semester. We have a few different types of stratigraphy that we can study as we introduce these basic stratigraphic approaches. One of the most important and basic ones is lithostratigraphy. In this case, we're actually studying the stratigraphy sequences of different time through rocks. Now, these may be time transgressive, which means your lithostratigraphy may not always be the same age. In this case, we're studying rock units. We have no implicit time constraint in that stratigraphic ranking of rock units. So when you're looking at a geologic formation, which is the fundamental unit of lithostratigraphy, it can be a range of ages. It's not to say that if you look in the Platteville Formation in one location, it's going to be the exact same age in another. A formation can be grouped into, well, groups, or a formation can be broken down into members, and members can be broken down into beds. For example, the Jurassic Morrison Formation has members such as the Brushy Basin member, the Tidwell member, the Salt Wash member. And each of those members can then be broken down into individual beds, whether it's a sandstone bed or a limestone bed, and so on. Another way of describing stratigraphy is in chronostratigraphic or geochronologic units. So remember, lithostratigraphic units is just looking at rocks. We're not looking at anything else. We're just describing layers of rocks and their relationships to each other. That's lithostratigraphy. Chronostratigraphy and geochronologic units deal with time in different ways. Chronostratigraphy or chronostratigraphic systems are time rock units. So take the rock portion from lithostratigraphy, but now we are going to apply some kind of time context to it. The system is the fundamental unit of chronostratigraphy, and we can break up systems into stages, or series and series into stages. And what we're looking at here is the geologic materials that have accumulated over a certain interval of time. Okay, so these are the representations of time. So if we pick a random geologic period, let's say Ordovician, okay, the Ordovician period, that's a block of time. But all of the rocks and fossils that were produced during that interval would be called the Ordovician system. We talk about those pure time units as geologic periods. Okay, so geochronology is things like periods and eras and eons or epochs, ages, subages, and chronostratigraphic units are eonothem, erathem, series systems, stages, and so on. So if we're talking about the Ordovician period is an abstract chunk of time. It's a duration of time. Regardless of what rocks or fossils we're looking at, it's from this point to this point in time. Chronostratigraphy would be everything that we have evidence for during that interval of time. So you can have an Ordovician period. How do you know you had an Ordovician period? Well, because of the rocks and fossils that make up the Ordovician system. So to recap, we have lithostratigraphy, which is just rocks. We have chronostratigraphy, which is time in a rock context, and then geochronology, which is just pure abstract time. To further differentiate geochronologic versus chronostratigraphic units, our geochronologic units are just, again, abstract time, and we describe these based on early versus late. So you can have the early Cambrian or early Miocene, or the late Miocene or late Cambrian or late Ordovician. Okay, this is how we talk about things with time anyway. You might refer to it being late in the day or early in the day because we're talking about a period of time, an interval of time. Now, if we're looking at the materials that represent that time, in chronostratigraphic terms, you would refer to them as upper and lower, upper strata and lower strata, which represent this time stratigraphic context. Okay, so the upper Cretaceous refers to rocks. Those were formed and deposited during the late Cretaceous time. Okay, so when we talk about chronostratigraphic units, those are tangible. Those are actual rocks and fossils, and we describe them in upper or lower, depending on how they're stacked. They correspond with early and late, respectively. Okay, so with those basic concepts under our belt, we're ready to really look at biostratigraphy. So if lithostratigraphy is just looking at rocks, regardless of time, and geochronology is looking at time, regardless of rocks, and chronostratigraphy is looking at rocks in a time context, biostratigraphy is looking at fossils. We're not really paying much attention to what kinds of rocks, though that can be important. We're looking at telling time based on fossils, a description of sequential rock units based on fossil content. So in this case, instead of saying this is a sandstone that correlates to this other sandstone over some distance, we're going to say this rock contains fossils A, B, and C, and that corresponds to this other rock over here that contains the same fossils. So this provides us with a relative time control. Without biostratigraphy, we would have no geologic timescale. We could have radiocarbon dates or just radiometric dates for rocks. But if we didn't have fossils in them, we still wouldn't be able to determine what period is what, because remember, geologic periods are defined based on fossil content, and that's where biostratigraphy comes in. Geologic time and biostratigraphy are again based on phylogenetic evolution. It's that stepwise change in organisms over time, or populations of organisms over time. So one of the ways in which we describe biostratigraphic zones and ranges is what's called the range zone or the biozone. We define an organism's biozone, a fossil's biozone, by its first appearance, which represents loosely its speciation, at the bottom, and then at the top in the uppermost strata, we would see its last appearance datum, which represents its extinction. So every organism that has ever lived appears at some point and goes extinct at some point. That is its range zone, the time in between its first appearance and its last appearance. Now we have lots of different types of range zones. We can have a straight up range defined zone, which includes concurrent range zones. So if you're looking at two fossil taxa, and during the time in which they are concurrent or overlap in time, you can have a taxon total range biozone. This one species, when did it start? When did it go extinct? A partial range zone, which would be when this one organism lived by itself before something else evolved next to it. And then the interval biozone, the time in between two different species. So for example, the time in between hominids and Tyrannosaurus rex, there would be an interval zone there. Now, usually we want to do this on more than just one or two fossils, so we would use what's called an assemblage zone. And this is based on 3 or more coexisting organisms, coexisting taxa. And again, we define this based primarily on first and last appearance data, but we can also contain some of those other supplementary data like partial and interval ranges as well. We also have this phenomenon that we call abundance zones. This may not be the beginning or end of a fossil taxon. However, it does demonstrate when they became widespread or abundant. So in other words, we may have had a species that was present but was minor, and then all of a sudden later on it becomes very, very abundant. That's an acme zone, an unusually large concentration of a certain taxa. These we have to be careful with, because they could represent just a local phenomenon, or it could be multiple phenomenons on the local scale that gives it a more global appearance. So these can be useful, but we have to be very careful with how we use them. So here's some examples. Let's say we have a series of strata, and we're going to look at the unit at the bottom, the unit at the top, and the sliver in between. For species A, a total range biozone would be that its first appearance is at the margin of time 1, and its last appearance is shortly after time 2. A concurrent range biozone would be species B and C. B started out sometime at the bottom, and it goes extinct at the line for time 2. C starts out at time 1 and then persists beyond the scope of this observation. But notice there's a brief period in between time 1 and time 2, in that middle sliver where both of them are coexisting. That's a concurrent range zone. A partial range zone, like we see with D and E, is when D starts out at time 1 and persists through time. E starts out at time 2 and persists through time. We can describe the partial range zone of the time in between D and E coexisting. And then we have our interval zones where we're looking at specimens F, G, and H. You notice F and G start sometime in the past. F goes extinct at time 1. G goes extinct after time 2. And H starts out at time 2 and continues. So we could talk about an interval zone where this third taxa bridges the gap between F and H. So total range zones, concurrent range zones, partial range zones, and interval range zones are all different ways in which we can describe time among numerous fossils in an assemblage. Now, it's important to note that we don't want to use just any fossil. We want good biostratigraphic markers, and not all fossils make good biostratigraphic markers. Because we're relying on first and last appearance of certain species, we don't want something that's been around for 300 million years, because if we find a rock with that fossil in it, we have a range of 300 million years for what age of rock we're looking at, and that doesn't help us. So we want to go with something that has a short range, but is easy to identify and is abundant. We have a whole bunch of criteria for what makes a good index fossil. And actually, plankton, small microfossils, which is something we're going to get into in lab here pretty soon, really help out a lot with this. So small planktonic organisms with preservable hard parts are really the best. And there's a few reasons for this. Preservable hard parts meaning they can make good fossils. They're widespread. We find them all over the place. This is really important because if you want to do biostratigraphic correlation, which is the whole point of biostratigraphy, is figuring out time equivalence over distance, you want fossils that are widely distributed around the planet. It's not going to help you if you only find them in one quarry. 2, they're abundant. Plankton are very, very abundant. When I go out and I spend a summer doing fieldwork digging up a dinosaur, that is one specimen, and it's huge. However, with planktonic organisms, you can have a small little glass vial filled with literally thousands and thousands of specimens. So statistically, you want them to be abundant. You don't want them to be rare and hard to find. And second, small. Like I just said, if something's really, really big, it's hard to get a huge sample size. So if they're smaller and abundant and widespread, you can have really good statistical significance with your biostratigraphic analysis. Big fossils, as cool as they are, they generally make pretty poor markers. There's not as many specimens, and their stratigraphic resolution is not nearly as good as what we have with planktonic organisms. Furthermore, marine fossils are better for biostratigraphy than terrestrial. Again, they're more likely to be widespread because they're being transferred by currents throughout the oceans and widely distributed throughout most of the planet. They're less likely to be time transgressive because if it's out in the open ocean, you're not going to see a subtle or a sudden change in lithology. And they're much more likely to be of a continuous stratigraphic sequence. For example, the Atlantic Ocean has been accumulating sediment since its incipient rifting in the Jurassic period. We have a continuous sedimentary record, thus continuous record of plankton from the Jurassic to today in the Atlantic Ocean. We cannot say that for anywhere in a terrestrial setting right now. So also in general, the further back you go in time, the worse your stratigraphic resolution is going to be. Indeed, the further back in time you go, the fewer rocks you have, right? We have more rocks from the Cenozoic than we do of the early Paleozoic or of the Precambrian. Okay, so when we look at more recent material, we have a better biostratigraphic resolution. However, sometimes this is just a time of high sea level yield, which is giving us widespread datable deposits. So we have to be kind of cautious with what we're doing. And this is totally fine. You just have to be aware of the parameters as we're going through a biostratigraphic analysis. Here we can see an example of one of these range charts. So each line represents a different species of plankton. And you can see the ages going down on the left in millions of years. So at the very top is modern and at the bottom is 20 million years old. You'll notice that some of these lines continue off of the page more or less, and that means that they still exist today, and other things went extinct before. And you can see when things started. So the lower boundary of each line is its first appearance datum. The upper boundary is its last appearance datum. And notice where some of these bars become wider, that would be abundant zones or acme zones. So we have maximum and total ranges and then average ranges for their abundance as well. Now there's a very special way of plotting biostratigraphic data. We can do what we just saw on the previous slide, where we just make a chart and we show them all next to each other so we can look at the overlaps. And that's very useful, and that happens all the time. However, we have another way of plotting biostratigraphic data that's called a graphic correlation or age depth plot. And this is something we're going to be doing in the lab, but I'm going to introduce it here first. We're going to be comparing two stratigraphic columns and looking at similar fossils, both when the first and last appearance data are for them. And by comparing them instead of parallel to each other, but we align them perpendicular on like an XY plot, we can actually start learning more about how we can use fossils to tell us relative time. Okay, we can get absolute or relative sediment accumulation, and we can also identify hiatuses, or when there was a break in the sedimentary record. So here's an example of an age depth plot. You notice that the horizontal axis is age and the vertical axis is depth. And we have different symbols. We have little squares and little triangles, and what those are telling us is the first and last appearance datum of various fossils. What kind of fossils? It really doesn't matter at this point. We can then draw a line among all these points and see if that slope of the line indicates sediment accumulation rates. So with time over age, or so with age, I guess, over depth, how much sediment are we accumulating? Over time, if you notice that your slope changes, that means there's a change in the rate of sedimentation. If you have a completely horizontal line, that means through time we're not changing depth, meaning that we're not accumulating anything, and that would indicate a hiatus or a moment of non-deposition. It could indicate a facies change. It could indicate an unconformity. So we're going to see how we can construct these here in lecture, and then it's part of the exercise that you'll be doing in lab. So here we have a couple of examples of how we do this. So in the image on the left, where we have a well or outcrop A, and then we have a well or outcrop B on different axes. Okay, well, outcrop B is on your vertical axis, and outcrop A is on your horizontal axis. Notice our symbols. The blue numbers are indicating some kind of faunal ID, so species number 100, species number 90, species number 82, and so The red squares represent the fossil top. In other words, this is the last appearance datum or its extinction, and the circle represents its speciation or first appearance datum. You'll notice we have a first and last plotted for each faunal ID. And then down on the sides and the bottom of our graph, you can see some blue bars that indicate where is the first and last appearance for species 80, 82, 85, and so on. But when we plot these perpendicular to each other rather than parallel, we see that there's a best fit line among these that's about 45 degrees roughly. And that would essentially mean that outcrop A and outcrop B were accumulating sediment at about the same time. We know that because at 45 degrees, that's getting equal sedimentation among both sides during the same interval of time that these organisms were alive. On the diagram on the right, we see something very similar, except in this case, we're looking at the depth of a well over time. And we're seeing changes in our sedimentation rate. So starting for the diagram on the right, starting from its lefternmost edge to the right, we see about a 45-degree angle and then a horizontal line. The horizontal line means that we're not accumulating anything. And then we accumulate some more and then nothing. And we can actually start, if we have absolute age dates for this, we can apply that time and figure out how many millions of years we were accumulating sediment and how much. So for example, in between about 92 million years and 86 million years, we accumulated 30 meters of sediment per million years. And again, we can do this over large stratigraphic columns. So in this case, you're seeing a time depth plot. So you can either do 2 columns or you can do time depth plots. Either way, it's the same process. And we can see a very detailed, very high resolution image of how the sedimentation rates varied over time at this location. Now, we'll see some more examples of biostratigraphy in lab, but I want to talk about our last chapter for this unit, which is chapter 9, fossils and paleobiogeography. So we're looking at the distribution, spatial distribution of fossil taxa over time. So we're doing an analysis of spatial distributions of organisms, and essentially what we're going to be doing is plotting and mapping the distribution of plants and animal species or communities over time and over space. So paleobiogeography is everything that we just set up there, this spatial analysis, but we're doing it with fossils. Okay. So a couple of terms that I want to get, again, under our belt for paleobiogeography is, first is a realm. A realm is a continental scale biogeographic region. So a realm would be something like a large scale continent, whether it's temperate or so on. A province is an ecologically defined realm. So a desert, a coast, a rainforest, some kind of boreal forest. And these are usually defined by animal populations and communities. We would define a desert province mostly by what kind of animals we have living in it. The realm itself is going to be like the desert, but the province is going to be defined as the organisms living in that desert. A biome is similar to a realm, but in this case, it's an ecological community characterized by what we call a natural region, and it usually refers to plants. So this would be rainforest, deserts, or tundras in terms of what kind of plant life is there. We do throw coral reefs in this area as well, even though corals are indeed animals. They have ecological roles very similar to plants, at least in a terrestrial setting. So these are often very strongly correlating with altitude or latitude, so think about climatic belts. So how high up something is. If you go from the bottom of a mountain to the top, you'll notice a whole bunch of biomic changes. Or latitudinal changes, going from the North Pole to the equator, for example. You'll see a number of changes in your biomes. Now, not all biomes are the same. Terrestrial biomes are usually very strongly influenced by temperature and moisture flux. So again, think about the difference between a tropical rainforest biome versus a desert biome or a tundra biome. Temperature and moisture play huge roles here. Aquatic biomes, we're talking about something like a coral reef, moisture isn't really an issue because it's underwater, but we still use temperature, but we also use chemistry such as salinity and the type of nutrients that we have there, oligotrophic, eutrophic, and so on. We can describe the populations in biomes, whether they're aquatic or terrestrial, based on characteristics such as whether it is endemic. This would be a native or restricted population. A cosmopolitan population would be more widely dispersed, and a disjunct would be a population that is present in more than one place but has gaps, so it's not continuous. Okay, so an endemic population would be we only find it on one continent. A cosmopolitan would be finding it on numerous continents, and disjunct would be we find them on 2 continents but not a third one that it could potentially live in, and we don't find it there. So these are different ways of describing populations within different types of biomes. So how do taxa get around the planet? How do we disperse different types of organisms? Well, the distribution of different taxa is influenced by a couple of things. Climate and environment, and that organism's method of being transported or distributed. So, for example, if it's an active organism, can it walk? Can it swim? Can it fly? So that's how it's getting around. Or is it a passive distribution? Does it float? Is it carried by wind? Is it carried by others, perhaps parasitic or some kind of commensal relationship? Okay. These are 2 different ways in which we can describe how organisms are actually distributed. Now think about climate and environment is going to play a role in that too. If it's an organism that is able to be passively transported by floating, then the environment, being an aquatic environment, is going to be useful. Climate also plays a role too. For example, if we're talking about the distribution of plants, climate, plants and then wind patterns are going to play key roles in this as well. And even in these methods, we can break them down even further. Airborne distributions. Pollen and spores are distributed by wind. Some seeds are carried by wind. Think of dandelion fluff or milkweed. Those are all seeds. Insects and arachnids are very, very important for plant distributions. Mold and bacteria can also get in the air. Anytime you have allergies, that's what you're experiencing is pollen and spores and mold and bacteria. And some organisms indeed are just migratory flyers. Ocean currents are very, very effective for our passively floating organisms. Plankton, any kind of benthic organism that has a planktonic larva. There's a number of organisms that may be stationary at the bottom of the seafloor in their adult stage, but as juveniles, they are planktonic and freely float. And we have things that are rafted biota, plants, animals, and fungus, things that can be rafted on logs. Here's an example. Think about the cartoon image of a desert island. Okay, a little island, just sand, and there's a coconut tree sticking out of it. How the hell did a coconut tree get there? Well, because a coconut probably rafted its way and just floated up onto that spot of land and grew. Okay, so these types of rafted biota are very, very important as well. So these are all just different ways in which we can distribute different types of organisms. In many cases, organisms are just carried by others, and this includes, in a way, what humans do when we introduce an invasive species. Parasitic organisms that include viruses, something we've been hearing about a lot, are carried from one organism to another and then distributed. We have commensal or mutualistic relationships, so remoras on sharks. Remoras stick to sharks and help clean parasites, and as a result, they're protected by the shark. Barnacles, which will grow on the bottom of ships or logs or even some other animals. Algae, juvenile gastropods that will get stuck in the feathers of ducks and geese and other waterfowl will then be distributed from one pond to another. And then we have our anthropogenic situations, like I said, with the planned or even accidental introduction of any kind of foreign plant or animal. In many cases, these have pretty devastating consequences. Around here, we have a problem with zebra mussels and emerald ash borers. So a bivalve and an insect that have been introduced, and we try to keep them under control. There's also a problem in the Fox River and Lake Winnebago of trying to keep certain type of lamprey larvae out. There's certain types of fish that we do not want in there. So whether it's intentional or accidental introduction of invasive species when we introduce something that is a form of being carried by at least our species from one location to another. Now, we can look at comparisons between paleobiology and ecological biogeography. The basic idea here is that organisms that have a very wide geographic extent and spread should be the ones most likely to subdivide into separate populations and then speciate what we call allopatrically. So allopatric speciation is a form of speciation where organisms are, one organism branches into two because they've been physically separated. So you have one main population, and there's some kind of physical barrier that causes that population to split in two, and they ultimately evolve in different ways. Okay, so we would think that if you have a wide geographic spread, that's going to be more likely. However, the method in which we distribute these organisms really dominates what we call population migration. So there's a couple examples here. One, planktonic larvae. Okay, so organisms that have planktonic larvae can disperse those into a constant gene pool because of mixing, and so then we don't really have a lot of speciation, and the fossil record does tend to show evidence of this. Another example would be gastropods that don't have planktonic larval forms would typically show a very slow dispersal pattern, but really high speciation rates. So this would be a combination of allopatric speciation and what we call sympatric speciation, where you have one population in one place and they just start doing different things and they separate into two species without a physical barrier. So we see a little bit of both of this in the fossil record. So here's an example of dispersal with the European periwinkle, which is a snail, a marine snail known as Littorina littoria. They're very common across northern European coasts. That's where their homeland is. And I want to mention this is a snail that does not have planktonic larvae. They actually have baby snails that look like the adults but smaller, so they're non-planktonic. So they shouldn't be widespread because it's a snail and they're slow. However, in the 1860s, they were first noted in Maine. And then they dispersed southward towards New Jersey by the 1890s, and today they're present as far as south as Virginia, and they are the dominant North American East Coast gastropod taxon. They're very, very abundant. They've somehow got over here from Europe and diversified and are really, really abundant. So how did this happen? They were introduced on accident to the U.S. from ships that were coming back and forth from Europe and America, bringing cargo. You see, when you have a ship going across the sea, you have to weigh it down enough where it'll actually float properly, and so you have to have that weight. If you have a bunch of cargo and you're coming from Europe to the United States or from the United States to Europe, you have to have that weight in the ship. So we think ships were going from the U.S. to Europe with cargo. When they got to Europe and they sold all the cargo, they needed to put rocks in the bottom of the ship to help weigh it down for a proper sail back to America. And this is known as ballast stones. However, when they were putting all these ballast stones in there, they accidentally grabbed some snails that were stuck to the rocks, and those took a nice long voyage to America, where those ballast stones were then taken out of the ships and new cargo was put in there, and now we have European snails in North America. But there was so much of this going on, they were really widespread and moving all up and down the eastern seaboard of the United States. One minor caveat to this is that we did find in Newfoundland at Viking archaeological sites, there are European periwinkle shells which may have been carried as food, but this doesn't seem to correlate to the widespread distribution around the 1860s to today. Interesting though, we could do comparisons of the DNA of our different Littorina populations, the ones in Europe and the ones in North America, and see if they've started to undergo allopatric speciation, if they've started to take on a different genetic code because they're in drastically different areas. Now, we can also use biogeography and paleobiogeography and evidence of climate change to make some analyses as well. If we look at the migration of some species during glacial or interglacial times, whether it's a 41,000 or 100,000 year Milankovitch cycle, we can see changes in pollen spectra in lake sediments. We can see changes in tree lines from northern, southern, or in vertical sections. and also look at how different marine plankton will transfer and function throughout time and space. Sea level changes will influence very short-term allopatric speciation and allopatric distributions. For example, if you have a sea level fall and you can now get across Beringia from, like, Europe and Alaska, that would be a temporary change, because when the sea level goes back up, that migration route is going to be broken off again. And we also have this concept of refugia. Refugia is when you have an organism that's thought to be extinct, but it's actually thriving in one small population in one area. Think the famous fish, the coelacanth, which people thought were extinct from the Cretaceous, but actually there's a few living off the Indian Ocean and Sulu Sea. So think about, when did the last tree grow in Antarctica? Okay. At one point, Antarctica had forests. When did that stop? And this could be a response to, in terms of modern climate change as well. The reestablishment of grasses that we're starting to see on parts of the Antarctic Peninsula is another indication of a radiational change happening because of climate. So, for example, we can look at some sedimentary records. In this case, the upper chart, the colored chart there you're seeing, is from Chatworth Bog in Illinois. If we look through that bog and we look then in years before present, so it goes from 1,000 years ago all the way to 14,000 years ago at the bottom, we can look at what different types of pollen we find over this 14,000 year record from the Pleistocene into the Holocene. And what we find is, in this same location, 14 to 12,000 years ago, we had lots of spruce and then ash. And then as we go up in time to more recent, we see an influx of elms and a reduction of spruces and ash. And then around 9,000 years ago, we start seeing more grasses, ragweed and goosefoot, and some sages. Oak trees start to diversify around 11,000 years ago and stay pretty consistent. But notice that we lose spruce, ash, and elm trees as we start to gain grasslands, ragweed, and goosefoot. We can also look at changes over the last 2,000 years in New Zealand. Now, this is due to human influence vegetational changes. Forest versus open shrubland. Again, in this case, we're looking at depth, which does correspond to thousands of years. But you can see a shift from a forest to an open shrubland as you go down into the depth of these cores, indicating some massive changes around 1850 or around even in sometimes going back hundreds of years. And finally, what about the role of plate tectonics and biogeography? You can have organisms living on one chunk of crust, but if that chunk of crust is moving around over hundreds of millions of years, you kind of need to be able to trace that. This leads to very long-term allopatric speciation. Again, you're separating a boundary. If I have one population living in an area, and that area rifts in two, and now I have half the population on one continental mass and the other population on a different continental mass, those are going to genetically drift away from each other to a point where they're now unrecognizable species. Okay. Now think back to the Permian period when we had Pangaea, the supercontinent, a world with one giant ocean and one continent. We probably wouldn't have very many distinct provinces or realms. There's not a lot of physical separation and barriers. There's some, but not nearly as many as today. We have all these continents spread around the globe today, separated by oceans. Back in the Permian, we had one giant continent. So it was much easier for organisms to get from one side to the other. Okay. When you have continental separation, this leads to what we call provincialism. This is part of that speciation. And this can lead to also what we call diachronous distribution. So if isochronous is one time, diachronous is two times. So a distribution over two different times of taxa over large distances. This produces really wide geographic distributions of species, and it probably will enhance the survival of that lineage by having it widespread. It's less likely to suddenly go extinct from some local event. But it is something to keep in mind that fossils do continue to move after death if they're on a continent, and that continent has been moving around due to plate tectonics. In fact, fossils are a very important part of our understanding of plate tectonics. Paradoxides pinus, this famous trilobite from the Cambrian in Europe and in North America, was identified by Alfred Wegener and many others as an evidence of these two rocks and these suspect terrains are the same age because they have the same fossils. Even though these continents are now thousands of miles away from each other, they have the exact same fossils in the rocks, so thus they're about the same age. And that's the end of the material for Exam 1. So Exam 1 will launch on Canvas on Monday, October 12th, is when we will have our first exam. It will cover content from lecture and lab for weeks 1 through 5. Okay. So it will include fossil record and taphonomy, ontogenetic variation, taxonomy, systematics, and phylogenetics, evolution and extinction and functional morphology, paleoecology, biogeography and biostratigraphy, and micropaleontology, which we'll be covering in lab this week. Obviously, the exam is going to be open book, open note. Realistically, that's the best way to do it right now. However, answers should be thorough, and I encourage you to use cited outside sources. Okay, so when looking for examples, which it'll commonly ask for examples, find some good examples. When you've completed the exam, it should be submitted to Canvas as a Word or a PDF document so I can easily open it and grade it. And the exam is due by October 16th, so you will have the week to do it, and it's a hard deadline. I'm not going to accept anything after the 16th. So pace yourself through the exam, but don't lose track of time. Make sure you manage the time efficiently, and make sure your answers are thorough to the best of your ability. And if you have any questions, please feel free to let me know. So we'll see you next time.